Hey fellow scientists! Today we're going to be talking about DNA, but first we have to start off with the basics. What is DNA? Well, deoxyribonucleic acid, better known as DNA, is a molecule of heredity that provides instructions for the cell functioning. It is also the blueprint for the production of proteins that do the work of the cell. DNA is not usually visible to the naked human eye, but today we're going to get a close-up look at DNA from a kiwi.
Michael, that's the wrong kiwi.
What? Close-up look at DNA from a kiwi. To conduct this experiment, you will need a kiwi, dish soap, salt, and water. And let's not forget about the knife and alcohol, because those two go so well together. The first thing you want to do is put your alcohol in the freezer for 30 minutes before you start your experiment. I'm back. Next, we need to peel the kiwi and dice it into small pieces. Peeling the kiwi helps extract DNA much easier, and the skin doesn't even have that much DNA anyways. After, you want to place your diced kiwi in a bowl and smash it with a fork. Smashing it helps to break up the cells of the kiwi so we can extract our DNA. Now set your bowl aside for now. Now it's time to make a mixture that breaks up our cells even further to release more DNA. Mix 2 grams of salt into 100 milliliters of water. Next, add 5 grams of dish soap into your mixture and stir gently so no bubbles appear. This is called an extraction buffer, which means it breaks up the cells even further to release more DNA. Now bring on back your smashed kiwi and add your solution. After you're done with that, place your bowl of kiwis into another bowl of warm water. And then set aside for 15 minutes. But all jokes aside, guys, let's talk about something that makes you, you. Something that was brought down for generations. Let's talk about your DNA. But specifically, the structure of DNA. I went there. DNA is not just one single strand, but consists of 2 nucleotide strands that twist together and run in opposite directions to form a double helix. It is held together between hydrogen bonds between bases. It's really not as complicated as you think. Each strand of DNA contains 4 parts of the nucleotide. Each nucleotide contains deoxyribose, which is a 5-carbon sugar. They are bound to one side of the phosphate group, while the other side is bound to the nitrogen-containing base. There are 2 different groups of nitrogen bases, the purines and the pyrimidines. Purines are a double ring structure that contain the bases adenine and guanine, while pyrimidines are single ring structures that contain the bases thymine and cytosine. Certain bases can be binded with another to form DNA. The base pairing rules are adenine with thymine and cytosine with guanine. Next, we're going to talk about DNA replication. I went there again. So DNA replication is a process of DNA copying itself exactly. The structure of DNA can explain how DNA can replicate. Hydrogen bonds hold DNA and are easily broken apart. Then one of the broken strands serves as a new template for the new strand of DNA. The old strand of DNA that was previously broken apart remains intact. That's why we call DNA half old and half new. The replicating process will go on, and the new strand will eventually become the old strand, and so on. Enzymes basically push along this process. Helicases, which is a type of enzyme vital to all living organisms, unwind the double helix. Fun fact, the area of DNA that is opened by DNA helicase is known as a replication fork because it looks similar to a fork in the road. Anyway, then DNA polymerase, which is an enzyme, attaches to the building blocks for DNA, which is nucleotides. DNA ligase, which is another enzyme I might mention, fills the gap. Do you see the common denominator here? They're enzymes! Not only to mention that enzymes wind the double helix back together. So where was I again? Yes, replication. Now, this process always doesn't go so smoothly. Mistakes can happen. But when they do occur, glycosylase removes them. They are replaced with the correct base. But that's besides the point. You may be asking yourself, why and when would this ever happen? The answer to your question, my dear, is the S phase, or the second gap phase. To be more exact, it is a portion of the cell cycle interphase. In fact, the S phase is the only phase in which DNA is replicated. Here, the DNA is divided and passed into the daughter cells. Now, daughter cells are one of the 2 cells created when a cell undergoes cell division. They have the same genes that are identical to their parent cells because they contain the same number of chromosomes. Oh. Well, our mixture is unsettling. Let's take a look. Now what you're going to want to do is get one of these things and place it over your cup. Just strain the mix into the cup and set the remnants aside. You won't be needing those. This green liquid contains our kiwi DNA, but the question is, how do we get to see it? With alcohol, of course. Now pour your alcohol gently down the side of your glass. You'll notice that the alcohol stays on top while the kiwi mixture stays at the bottom. This is because the alcohol is less dense than the kiwi mixture.
These tiny white strands that start to form represent our DNA.
Congratulations, you can now observe kiwi DNA with the human eye. Fascinating. I'm so sad to say, but our experiment for today is all done. It was so great hanging out with you today in my kitchen. Maybe we'll cross paths one day, have a cup of tea, or who knows, see you in a 9th grade biology class. I can't wink, so I'm gonna just leave you with this.